Bismillahirrahmanirrahim. Assalamu alaikum everyone. So today we will discuss what is cholinergic transmission. So you know that you have this type of neuron, and on this neuron at this terminal ending or its nerve terminal, you can have certain amount of neurotransmitters released. So if this is your terminal button and this is the postsynaptic membrane, so you have this presynaptic membrane and this postsynaptic membrane, and this whole thing is the synaptic cleft. So two types of neurotransmitters can be released, and on the basis of this we classify the receptor or the transmission. So if the neurotransmitter that is released is acetylcholine, we call this cholinergic transmission that we are going to discuss today. But if the neurotransmitter that is released is norepinephrine, we call this adrenergic transmission that we will study some other day. So now we will talk about acetylcholine receptors, or type of the transmission that involves acetylcholine as the neurotransmitter. So now these cholinergic substances or these cholinergic nerve endings, where are they present? They are present in autonomic as well as in the somatic system, but both of these are in the efferent system. Like in the autonomic efferent preganglionic portion, we have this cholinergic transmission happening. Similarly, in the somatic efferent to the skeletal muscles, we have this cholinergic transmission. So it is both in somatic, whereas in the autonomic system, in the somatic one we have it in the efferents to the skeletal muscle, whereas in the autonomic one we have it in the efferent that are preganglionic. Now they are saying that if this is the nerve terminal, this nerve terminal has these vesicles. The vesicles that are closer to the membrane or closer to be getting released, they are small, they are more in number, and they are transparent, and they have acetylcholine in them. Whereas the ones that are farther away from the terminal, a bit over here, they are large, they are dense, they are also very few in number, and they contain peptide co-transmitters as well. So the ones that are small, that are clear, that are closer, that are larger in number, they are acetylcholine, they are containing acetylcholine, whereas the ones which are large, they are dense, they are less in number, farther away, they contain peptide co-transmitters. Now, when this is the whole neuron, if you know, this is the neuron cell body, this is the whole type of axon, these branches are dendrites. So they are saying whenever we are going to discuss the cholinergic transmission, we will talk about the synthesis of the neurotransmitter. So this acetylcholine neurotransmitter, it is being synthesized in the neuron cell body, this portion, this neuron cell body, or sometimes it can also be synthesized in lymphocytes. Main area is this neuron cell body. Now transport, because it has to travel from this cell body to this terminal button, so we have to do the transport through the axon. So this type of transport is axonal transport. Synthesized in the cell body, goes to the axon terminal, so the transport is axonal transport. After that, we talk about the vesicle. Now this vesicle, the two types of vesicles that we've discussed, the vesicle that is now transparent, how it is standing over there, or how and why it is nearer to the cell membrane to be released. It is nearer because it has this type of snare protein, fusion protein. This is the vesicle associated membrane protein. Now this, because it is present in the vesicle membrane, so it is vesicle associated membrane protein. What is it going to do? It is going to align these acetylcholine vesicles nearer to the membrane to be released. First is alignment, second one is, and it also triggers the release. First alignment, second triggering the release. So it has two works. Now this VAMP, the vesicle associated molecular membrane protein, this vesicle associated membrane protein, it attaches to this type of another type of receptor, another type of fusion protein, that is SNAP. Now what is SNAP? That is synaptosomal nerve associated protein, because this portion is nearer to the synapse and also it is associated with the nerve, so we call this synaptosomal nerve associated protein. The VAMP and SNAP, they are fusion proteins, they connect with each other. Because of their connection, they allow the alignment, second they trigger the release of the vesicle. Now this was the whole phenomena regarding this. Now they are also saying that why does this neuronal firing of the acetylcholine is very fast. This ultra fast neuronal firing is because the recycling of clathrin coated vesicles from endosomes. What happens is that whenever there is some exocytosis and this vesicle is released into the synaptic cleft, there is rapid clathrin coated vesicle recycling. So this vesicle is recycled again very rapidly by this clathrin coated protein. That is why they have ultra fast neuronal firing. The membrane has this rapid recycling capability with the help of clathrin protein. Now we will go to this very nice type of diagram that is given in Big Cadson. Let's have a look. So we have this diagram over here. So now this is the terminal button or the terminal knob of the nerve, and this is the synaptic cleft, and this is the postsynaptic membrane, and as I told you, this was the presynaptic membrane. So now what happens is that we have to make acetylcholine, and that acetylcholine, it is synthesized where it is synthesized in either the cell body or in the nerve terminal. So now they are saying this acetylcholine, it is formed from two things. One is acetyl coenzyme A and other is choline. Now this acetyl coenzyme A, it is present mainly, or it is formed from mitochondria, whereas this choline, it comes from extracellular fluid. This extracellular fluid, how is this choline going to enter the neuron or the nerve terminal? It is going to enter by means of this symporter. Symporter is a type of transporter which transmits both the molecules in the same direction. So this symporter, that is choline transporter, this symporter, it is sodium dependent. It transports sodium as well as choline from the extracellular fluid to the interior. So now acetyl coenzyme A from mitochondria, choline coming from extracellular fluid by means of this choline transporter, that is sodium dependent. Now both of them combine together by means of this choline, this CHAT enzyme. This CHAT enzyme is also known as choline acetyl transferase. This choline acetyl transferase enzyme then combines these two and forms this acetylcholine. But now this acetylcholine will not be present like this, rather it is going into certain vesicle. When the acetylcholine goes into vesicle, it is transported by means of an antiporter. Symporter was this type of molecule which was pulling both the ions towards the same side, whereas the antiporter, it pulls one ion to the interior, other to the exterior. So this antiporter is pushing the hydrogen out and allowing the acetylcholine to go in. Now as we know, this acetylcholine goes inside the vesicle, and what happens? It is now stored in the vesicle. Now whenever it is stored in the vesicle, how and why it is stored? It is stored because it has this acetylcholine itself is positively charged quaternary amine, and it is stored here, or it is having an interaction with a negatively charged vesicular proteoglycan. So this is how both of them, they have a positive negative interaction, and this acetylcholine is bound inside that vesicle. Now what happens whenever there is going to be action potential? This action potential is going to come in, and this action potential will allow this N type of calcium channels to open. When these N type of calcium channels open, they allow calcium to come in from extracellular fluid to the interior. When the calcium comes in, what happens? That this calcium allows these type of VAMPs, both of these vesicular associated membrane proteins, to allow this acetylcholine vesicle to open. Now this vesicle opens, there is intrusion of certain cations. What are these cations going to do? There a pore is formed, but this pore allows the certain positively charged or cations to come in, and these cations, they will replace this acetylcholine from the vesicular proteoglycan, and the acetylcholine will be ready to be released. So up till now we studied acetyl coenzyme A from mitochondria, choline from extracellular fluid coming via sodium choline symporter. they come together, they are facilitated by this choline acetyltransferase enzyme. Acetylcholine is formed. It is entered into vesicle by means of an antiporter, that is vesicle associated transport protein. This puts acetylcholine inside, hydrogen outside. Acetylcholine is bound inside the vesicle because it is positively charged. We have this negatively charged something which is known as the vesicular proteoglycan, which holds it inside. Now whenever the calcium action potential will come, the calcium channels will open, calcium will be influxed. That is going to activate the VAMPs. Also there will be a pore is formed, intrusion of the cations, and the vesicle is released, and the acetylcholine is freed from that proteoglycan thing. Now acetylcholine is released into the synaptic cleft. This acetylcholine can have two fates now. It can either go to these cholinereceptors, or it can either go to this cholinesterase. Cholinereceptors, whenever they will attach this acetylcholine, they will produce a response. Whereas when this acetylcholinesterase is there and acetylcholine goes to that, what will happen? This cholinesterase will break it into choline and acetate. So two fates, either attached to cholinereceptors or be broken down by acetylcholinesterase. Up till now, we are very much done with the whole type of transmission, etc. But we need to recognize certain drugs that can inhibit the activity of these transporter. So first drug is hemicolinium, or halt the colinium, or you can say it will inhibit the entry of the choline inside the nerve terminal. When it will inhibit the entry, no choline will be formed. So CHT you can say blocker is this hemicolinium, halt the choline. Similarly, you have another one which will not allow the acetylcholine to be captured in this vesicle, that is besamicol. Name is coming from vesicle, so besamicol. CHT, the choline transporter, it was inhibited by hemicolinium, halt the colinium. The vesicle, to prevent acetylcholine to get into the vesicle, we have this besamicol. Similarly, we have another one which is going to block this exocytosis, and that one is botulinum toxin. This botulinum toxin, it either breaks down these VAMP or these SNAP proteins. The VAMP ones, as I told you, associated with the vesicle membrane. The SNAP one associated with the presynaptic membrane. It can do the enzymatic cleavage of any of these and hence does not allow the acetylcholine to get released. So we did three drugs. For CHT, we got hemicolinium. For to not form the vesicle, we have besamicol. To prevent exocytosis, we have botulinum toxin. So these were the three that we need to discuss. Now they are also saying, in the text it is written that there is this tetanus toxin. This tetanus toxin has similar activity of enzymatic cleavage of either the VAMP or SNAP proteins, and it is an inhibitory neuron. It is present in the inhibitory neuron. That is why whenever you get tetanus toxicity, you get muscular spasm. Now they are saying that whenever there is this action potential comes, whenever there is depolarization, several hundred quanta of acetylcholine are released into this synaptic cleft to be attached on these cholinereceptors, or either to be broken down into choline and acetate by these acetylcholinesterase enzyme. Now we also have this acetylcholine can also have certain other fate. That other fate is attached to the presynaptic receptors themselves. They can either be acetylcholine. So this is the presynaptic membrane. This membrane can also have receptors. They can be acetylcholine receptors or heteroreceptor. Hetero means for a large amount of products. So it can either be attached on postsynaptic receptor, the cholinereceptor, either broken down by this acetylcholinesterase enzyme, either go to these presynaptic receptors, that is acetylcholine receptor or heteroreceptor. Up till now, I guess you are clear with all the type of drugs, the type of everything, and we have also discussed termination, how it takes place. Now we need to discuss certain other names of certain other products for your information. One is this one. So they are saying that its vamp is a type of snare, and we call this snare protein because the fusion protein, they are always known as snare proteins. So this vamp is a V snare, whereas snap is a T snare protein, and both of these are fusion proteins. Vamp is also known as synaptobrevin, whereas this one, snap, is known as syntaxin or snap 25. These are the type of additional names that you need to know. Similar to that, we also have this, they're also saying that because acetylcholinesterase is going to break the acetylcholine, if it is present in large amount, it is going to decrease the short life of acetylcholine. So it will decrease the acetylcholine half-life. Very short half-life will be produced when the breakdown enzyme is more in number. Similarly, they are saying that this acetylcholinesterase may also be present in red blood cells, and liver and blood plasma also have a certain type of acetylcholinesterase enzyme, which is a pseudocholinesterase enzyme. Pseudocholinesterase. So up till now, till this we have covered the type of transmission that involves acetylcholine, and that is cholinergic transmission. We did 3 drugs: the hemicolinium, the visamicol, the botulinum toxin, and also tetanus toxin. I hope you understood each and everything that I tried to explain through this lecture, and I hope you are clear about these all things. In the next video, inshallah, we will be discussing adrenergic transmission. Please like, share, and subscribe to my channel, and write a nice comment below. And if you want to see more of these videos coming, let me know. Inshallah, we will make a lot of this stuff.